Okay, awesome. Naihua, you're amazing in terms of making a draw. First of all, you're just amazing.
Oh, thank you.
But you do draw a good crowd. So, Dallas?
Well, we have fortunate to have Naihua here with us today, who several of us in the room, many of us in the room, worked with over the years, and I personally am really grateful to have been sort of had a lot of my thinking that's informing my work now shaped by working with Naihua in the past, and in particular on this sort of N-of-1 patient trial stuff we're going to hear about today. Naihua has an illustrious career that I don't know all the details of, but he's just retired from Columbia University, is that correct?
About a year ago.
About a year ago. And now doing fun things like going to NIH meetings and traveling and...
Lake Como.
But I think Naihua was quite influential in many fields and has been a visionary thinker. And so I'm looking forward to hearing his presentation today.
Okay, thank you, Dallas. Well, I'm very glad to be, almost feels like back home. I had a really good time in this room in the years past when I was at CCH. And very glad to see my many friends I knew before, and some we haven't met. Look forward to meeting you. And so Dallas, if I have some influence on you, I'll try to influence the group in some out of the left field way, and you'll judge at the end of the talk. So it's a long title, so I say a few words about the title. So the methodology I'm talking about is single patient trials, and it's also called N-of-1 trials. And most of my colleagues who work in this area use the N-of-1 terminology, and but it's not a good brand name for marketing. So single patient trial, I think might be a more appealing way to present the methodology for broader use. So I explain later why it's dealing with clinical decision in a pragmatic way, and it's importantly patient-centered. So let me see if I can get this to work. Okay, so there are two closely related themes I want to talk about. The first part is what the title says. I will talk about the single patient trial and try to share with you my thoughts why it is a useful tool for clinical decision making. And then, as much as time allows, I'd like to talk about some broader issues in the second part of the talk about using local investigations to inform local decision making, like for a implementation project or quality improvement program. And along this line, single patient trial is, I guess, one of the maybe exemplars for such a local investigation. So I will come back and try to talk about those broader issues later. Notice that I use the term local investigation, not local research, and I will try to maybe persuade you to think about ourselves as investigators, not researchers, for reasons I will explain. Okay, so the outline, the first part, I will give a brief overview of the single patient trials. Some of you might be already familiar with methodologies, but just to make sure we're on the same page. And then I will talk about a couple of key implementation issues, how this methodology needs to be implemented. And then I will go to the second part to talk about local investigation and local knowledge, and then discussions. Dallas, can you let me know when like I'm at about 30 minutes?
Sure.
So leave about 15 minutes. So okay, so the clinical question that a single patient trials tries to address is for patient with a chronic condition, which could be chronic pain or could be HIV positive status, and that's a chronic condition that is not going away. And so how do we take care of those patients? And it happens often that there might be multiple treatment options, and it's not clear which one is the best approach for this specific patient. Sometimes it's because like the various combination therapy have not been tested in research studies to inform the choice among the different treatment options. And sometimes there might be a treatment that has been studied in the past. There might be some existing knowledge about the average treatment effect, that maybe A is better than B on the average. But there might be, for a lot of treatment, a substantial heterogeneity of treatment effect. So the same treatment might not work the same for every patient. A might work better for some patients, and for some other patients, B might work better. And that we might not know. And so if a heterogeneity of treatment effect is an issue, very often we don't really exactly know what is the best approach for a specific patient. So one possible solution is just to do a investigation on the specific patient to find out what works better for that patient. So that's a single patient trial. So the basic protocol for single patient trial is to do multiple crossovers. So if we have the two treatment options, A and B, and we put a patient on the trial to try on both A and B. And this is probably something the good clinicians will naturally do in real practice, but maybe in an informal way. And this is just trying to do that try and error in a more systematic way to try to reach a decision that's more informed. So we assign time intervals, let's say just say weeks. So 1 week the patient gets A, the next week gets B, and then the next week maybe stay on B, the next week switch over to A, etc. And the assignment can be randomized or does not have to be. It could be using counterbalancing. I'm not going into a lot of details on the pros and cons, but the idea is that we put patients on the competing treatment options in a systematic way, so we get some data under both conditions. And so we collect outcome measures at least every week, so every time interval, so we get some outcome under this A, under this B, under this B, under this A, etc. And ideally, if it's not too much burden, it would be better to even collect multiple measurements within the same week, so we get better information. And then we compare the outcome. So we collect the outcome under the A weeks and the outcome under the B weeks and compare to see whether the patient overall is doing better under A or under B, and use that to inform a treatment decision. So as a basic idea. Is that clear, or are there any questions? Okay. So some of the key features, or maybe I was thinking about saying pros and cons, but it's not totally clear what is pro and what is con. So the first pro or advantage of this approach is that I believe it is reasonably consistent with the naturalistic clinical practice. You know, when faced with uncertainty, a lot of good clinicians will try the different treatment options to make the decision, but usually not in a very systematic way. And all this is doing is to try to do that try and error in a more systematic way. So I think the attraction of this approach, it is a pool. So take what people are already doing and try to pull it up a little to do it better than what they are currently doing. And I think that's usually much more doable than to try to push something down to say, you got to do this. And there's a potential, if it works out, that this might improve the outcome compared to patients who are treated in the usual care with the usual try and error. And whether this claim is really true or not really needs some rigorous empirical evaluation. And I'll mention briefly a study that I'm working on with several colleagues that is trying to do that. And in order for this to be successful, there needs to be development of supporting infrastructure to do the IT work, to do the statistical work. This is something that potentially, like Dr. Bob Lebowitz could be interested in doing in his clinic with his patients, some of his patients. But the Bob might not have all the tools needed to set up the data system to do the analysis. So if there are some tools that can be developed and become available to Bob, there's a better chance he might do it. And so this is also part of the work we're doing in that studies I will come to later. And I guess what is potentially promising is that in recent years, mHealth technology has been jumping and becoming, I think, almost universally accessible. So the infrastructure that can be developed for people to access the trial instructions and procedures through their smartphones, through apps. I should have almost forgot. So another question that needs to be resolved is there needs to be financial mechanism for the providers to get paid for doing all this work. Okay, so this PREEMPT study I'm working on with several colleagues. Some of you might know Richard Kravitz at UC Davis. Richard and I have collaborated on several papers on single patient trials, although Richard insists to use the term in one trial, so we're still friends. So we're very excited. We got funded last year from the National Institute of Nursing Research for this pretty large scale study that we call PREEMPT. So what this study does is try to do single patient trial using mHealth for patients with chronic pain. So this is dealing with pain management, and there are multiple treatment options, and it's very often not clear to the clinician what is the best way to proceed for a specific patient. So we believe the single patient trial could be useful for a lot of the clinical decision situations. So the first part, there are two phases for the study. The first phase is to develop the infrastructure, the IT system, and the statistical analysis utilities. That is ongoing right now. And we're hoping to finish the infrastructure work and probably starting sometime by the end of the spring to begin recruit patients into this randomized trial to compare us. So we will be randomizing patients to receive the single patient trial offer. They might not agree to do it, but they're offered, versus patients who did not receive that offer and just will be treated under the usual care to try to see whether the patients offered the single patient trial actually are doing better in the outcome later on. So it's very exciting. There are a few randomized studies comparing single patient trial versus usual care in the past, but I know of two that were published, and those are small studies and not very thoroughly looked at. And ours is probably the largest study so far to deal with this issue. Okay, so that's the overview. I hope you got some idea if you haven't had much experience with single patient trial in the past. Are there any questions, anything that's not clear? Okay, so I move on to a couple implementation issues, how this methodology could be used. So the first one is, this is not a panacea for everything. So there are conditions for which this is potentially useful, and there are conditions for which this is not appropriate. So there's a paper I published with two colleagues last year in Journal of Clinical Epidemiology, and the citation is in the announcement and also later in the slide. And there's a table that lists all interpretation for the indications and contradictions for this methodology. So the first indication is chronicity and stability. So in order for this methodology to be useful, the condition needs to be chronic. So this is a condition that the patient will have for a long duration of time. And also the treatment effect should be relatively stable. So it cannot be something that might work today and then totally become useless tomorrow. So under those two conditions, there's a possibility to learn from the treatment experience today to inform the treatment decision tomorrow. And there needs to be personalized knowledge because there's not enough already to tell us what's the best way to treat a patient. And then it's desirable, but maybe not totally necessary, that the treatment has a pretty quick onset when started, and then also a pretty quick extinction when stopped. So that when we go from A to B or B to A, that the switchover time, we will know the patient was responding to A during the first week, and A goes away and B gets started, and the second week the patient is responding to B. So for treatment that takes a long time to taper off or takes a long time to ramp up, it would be hard to tell whether it's A or B that is going on and what is going on. So this is a desirable feature, but if there is some transition going on, there might be possibilities to use statistical modeling to model that transition. So it might not be completely lost. On the bottom, I listed some of the examples of conditions that have been studied in the past with single patient trials. I have a kind of an open question that I'm ignorant about, that we were talking about yesterday. To what extent this type of methodology could be useful for treatment decisions for HIV positive patients, like to compare the effect for the specific patients on the different combination therapy, the different cocktails. I don't really know whether the response, like the viral load, responds very quickly or takes a long time to respond to a change in the treatment. I don't know, do we have a good clinician in the audience?
I think just by basic background knowledge about treatment options, I think the example you gave about A.A. So for people who are on antiretroviral therapy, I think if the treatment A fails, if you have failure on treatment A, that means the virus is developed resistance, so technically you won't be able to go back to that regimen if you're trying out a new regimen. So I think the proposed going back and forth between regimens may not be a viable option.
Okay. So in a sense, there is a dynamic long-term effect. Yeah. So one thing I forgot I should have mentioned that this is not something that is useful for treatments that have long-term effect, like surgery. Once the surgery is done, there's no way to go back to Plan B. But I would think that potentially there might be uses for this methodology in like behavioral interventions. There might be a variety of ways we can work with the patient to try to get the patient's engagement, and it might be possible to switch back and forth to see how the patient responds.
For ART adherence, I think there's like various behavior interventions that would supplement or enhance adherence to a regimen. So it's not the regimen per se, but it's different types of integrated combination prevention approaches that you can try with different patients.
Yeah. One point that might not be obvious is this does not necessarily have to be addressing the overall treatment question, treatment A versus treatment B. But this could be, a methodology could be used to deal with some nuts and bolt issues in tinkering within a treatment regimen that's doing well, maybe two different ways to explain something to the patient. So instead of evaluating the engine, a lot of time, part of the process of building a good engine is to evaluate the components, make sure they all fit together very well.
So
Yeah.
what about combination pharmacotherapies where maybe you start with a combination platform and then dismantle one of the arms off of the combination pharmacotherapy as the ABA? Would that work?
Yeah. That's definitely possible. Yeah. So the AB options could be withdrawal of part of the. So the usual care might be a combination therapy involving 3 medications, and the B could be withdrawing or reducing part of the combination. So there's a question whether the trial should be blinded or open label, and there's, our research training from the biostat department usually is that we should blind whenever it's feasible. There are some situations for behavioral therapies you cannot blind, but ideally, if we can, we like to blind. But here, I think the single patient trial offers some subtle question to be thought about. The reason we usually want to blind the patient in the usual clinical trials, that is the parallel group trials. Some patients get A, some patients get B. The reason for blinding is that we want to produce generalizable knowledge to apply to other patients who did not participate in the trial. So for those applications, blinding is often necessary because there might be artifacts like expectancy among the patient, trial participants. Say maybe a lot of trial participants like round pills instead of square pills. And so if they are not blinded, then their response, at least self-reported response, might include that expectancy. But that expectancy might not hold among the future patients. Therefore, it's better to blind so the future patients get the knowledge that is not, in this case, you could say, is confounded by the expectancy. On the other hand, for single patient trials, we try to produce the knowledge during the trial, apply it to the same patient in the future. If the patient likes round pills instead of square pills today, there's no obvious reason why he wouldn't. He will change that preference tomorrow. So, so his response to this expectancy is going to continue in the future. So I think can be made that the expectancy really is part of the package that the patient is responding to, not just the chemical compound in the ingredient. So under that perspective, maybe blinding is not necessary and it's actually better to do an open label to incorporate all the contextual factors that goes into the treatment. I think that's the philosophy of the pragmatic trial for comparative factor research. So so I would say at least it can be made, the case can be made that for single patient trials, because we are applying the knowledge to the same patient down the road, expectancy or the same kind of artifacts might not be something to control through blinding. And also, if we do it as an open label, it's a bit much, the bar of conducting the trial is much lower. It's less logistics to deal with. Another question is dealing with this issue of washout. So in the literature, a lot of the single patient trials are done with a washout period between A and B. So to wash out A and then get ready for B. So raising the question that I don't think has been really explicitly raised in the literature in the past. So this washout period potentially could help with the extinction part of the transition, but it does not help with the ramp up part of the transition. So if it takes 2 days for A to wash away, and then takes another 3 days for B to ramp up. So we do A and then wash out for 2 days and then start B. But B does not take effect, ramp up until 3 days later. So we actually have 5 days of transition in the middle. And alternatively, if we start B as soon as A is stopped, then there will be an overlap during which A is extinction down and B is ramping up. And by the 3rd day, the patient is on the full dose of B. And so that might actually reduce the transition rather than making it into 5 days. And there's another question that I think needs to be considered for comparative effectiveness studies. So it's possible that we might be doing the kind of thing like Steve mentioned. There is something that is currently being given to the patient. It's working not too bad, but we might try to see whether an alternative works better. So we have we're comparing 2 active treatments. And with the washout, we're actually adding a 3rd treatment condition of no treatment during the washout period. And ethically, that seems to be problematic, that if the patient is not doing too bad and the A and B looks, we thought, might be promising, why should we insert this B during the 2 days the patient will suffer with no medication? And it's also likely to be not very attractive, appealing to the patient. So in order to do this trial, I have to go without my, what is it called, Aliyah, for 2 days. So it might not be a very practical way to do this. An alternative that some of my colleagues have been working on, and there's a literature, small literature that goes back into the 40s of using analytic methods. So we have A coming down and B going up, and if we have frequent outcome measures, maybe on a daily basis, then we might be able to model how the A is coming down and how B is ramping up, and use that to get good estimates on what the stable treatment effect is, instead of having to force the patient
now,
to go.
There might be 2 conditions in which, any kind of washout would be hazardous to the patient. So, for example, if somebody's on antiretroviral drugs and you don't really want treatment interruptions, even if you are going to try a new regimen, if the old one is working, you're going to say, well, let's just stick with it. So I mean, that's kind of one condition. It can be harmful actually to have treatment interruptions. The second is where the medication, you need the medication to reach a certain blood level, and you need to maintain that level, say in the case of antidepressants or something like that. So you can't practically, the tapering off of certain medications like Paxil or Wellbutrin is not necessarily an easy process, and something you don't want to put people through. So
or even, imagine chronic pain, people may not be wanting to have 2 to 5 days of uncontrolled.
Yeah, so there's some limits. I mean, I see what you're saying, I see the advantages, but I think, at least in the case of medications, it could be complicated and not necessarily beneficial.
Yeah, so in many situations, it might be better to do without the washout in between, and so to make sure the patient is covered in some way. That might not hold universally. There's one possible scenario if the 2 treatments have a problematic interaction, then maybe they should not be, the patient should not be exposed to both at the same time. But anyway, that's an implementation question that needs to be looked at carefully for the. Yeah.
I have a question, a basic question actually. Could choose to answer it now or after you finish the presentation. So, if your interest is to see what's best for that one patient, then you're analyzing that one patient's data, right? But I could see reason for combining a number of people who are each on a single patient trial, and that is to see, is A better than B for certain characteristics of
Mhm,
subjects.
yeah.
Is that also part of what you're doing?
So this methodology could be used for both and with different emphasis.
Mhm.
One possibility is maybe a clinician only wants to do this for his own patient. That's a legitimate application. On the other hand, a research group might find this to be a good way to design a clinical trial and focus mostly on estimating the aggregate effect.
Okay.
But by the way, you can also give the patient the information so she will know which is a better treatment for her, and so anywhere in between. So both there's a spectrum of how this methodology can be applied.
So for the trial that you're doing, comparing RCT patients versus SPT patients, I imagine the hypothesis is a broader one because it's not restricted to only one patient, right?
Yeah. So we are looking at the aggregate. So for the population of patients exposed to N1 trial, are they doing better than the population of patients who were given usual care?
Regardless of A or B.
No. So the primary question for that study is the methodology.
Okay.
Yeah.
Okay. You're not comparing treatments, you're comparing these two options of going
That's right.
that way
Yeah.
or that way.
Yeah.
Okay.
Yeah.
Thank you.
Yeah. So along that line, another attractive feature about this is this is probably the most direct way to estimate how the treatment effect might differ across subgroups, because we have the treatment effect estimated for each individual patient, although maybe not very precisely. So it's relatively easy to compare how the males versus females respond to this treatment. We just need to aggregate all the male responses or aggregate all the female responses, and it's relatively easier than to try to do the comparisons in parallel group trials.
Mm-hmm.
Okay, so I will be coming back to kind of that dual nature of the single patient trial later. Okay, so the infrastructure needs, I think I mentioned earlier already, we need to have the IT infrastructure available to handle the data collection, etc., and the statistical infrastructure to handle the data analysis. So this can be taken out the clinicians' workload, and can be done in the background, maybe with some assistant, so that the responsibility for the provider to use this methodology could be made more acceptable. Okay, so a couple of publications. So I published this paper with Richard Kravitz and Chris Schmid last year in Journal of Clinical Epidemiology. And then Richard and I co-edited this user's guide for HRQ this past fall. And so we assembled a expert panel to address a variety of issues that includes ethical aspects, the financial aspects, and the IT issues, the statistical issues, and also the engagement and training for the end user, for the providers to participate in such a study. So the HRQ is about to release this report through their Effective Healthcare website, and they told me a couple weeks ago that it's going to be turned over to the final production group last week, and I checked this morning, it hasn't appeared yet. So, sometime soon, hopefully. Okay, and then Gabriel's paper is a very nice review of the N1 trials that have been reported in the literature, and the several Zucker papers have looked at the methodology for combining across patients. And Larson's is a very nice editorial, and he was also one of the early pioneers in this area. And Richard and I and other colleagues have written a couple papers related to how potentially N1 trials could be applied. How much time do we have?
So that was the 15-minute mark.
Okay, so let me try to go over the second part pretty quickly, so we don't run into the time for the core meeting. I also hope we can have a few minutes for discussions. So I think single patient trial is one example of what I call local investigation, and this is as local as it can be. So we're investigating a single patient. Oh, I should have mentioned earlier, although I used the term single patient trial, it could be a single agency trial. So if those of us who are working with agencies, if the agencies are thinking about different ways to do things, they could, we could be making a single agency trial in the same way. So I got sidetracked. Okay, so, but I think there are many other situations, especially in the domain of implementation programs and the quality improvement programs, where it would be useful to have some local knowledge to resolve a local question. One of the questions that I've been interested in for a number of years is this question about needing to adapt an existing procedure in a practical implementation, and to accommodate the unique circumstances in a local setting. I guess where the strength is, what are the resources, and what are the difficulties. And every agency is probably different in some ways, so the solution probably is different from agency to agency. That's why I call it local knowledge. And as opposed to the usual knowledge we are familiar with in research studies, which usually aim to be generalizable, and actually that is the way the HHS defines research. So I think the usual way many of my friends think about local knowledge is maybe talk to the local informants and to find out about their preferences or what they already know. Usually, I think it's not a very empirical approach, and I think there is a notion that in the local setting, you cannot do a statistical evaluation because of what is called the small n problem. Let me see. I think, yeah, so I'll skip a couple slides over. So here the issue is that what if we want to work with a specific clinic about a condition that they're trying to make some improvement on, and they might only have, let's say one of my colleagues has been working on patients in the VA with diabetes comorbid with depression, a pretty challenging condition. How do you treat them? And there might be only 500 such patients in this clinic. And so the low case n, the sample size, if you think of the 500 as a sample size, is too small to do a standard 5%, 80% trial. Actually, if you want to have 5% power and 5% significance, two-sided and 80% power, the sample size required is 500. So by the time you're done with the trial, all 500 patients have been used. And but the key, I think, is this is not a small lowercase n problem. This is a small uppercase n problem. The 500 is not the sample size, but rather is the population size. So for the local clinic, The primary concern is not how the knowledge gained from my clinic applies to the 35 million patients out there with the same condition. My primary concern is how do I take care of my 500 patients? So that's a quality improvement question instead of human subject research question. So the 500 is the capital, is the population size, and there is a statistical literature in the 60s that I learned when I was a graduate student, but then completely went to the back of my head because I never had a chance to use it or see it used. The literature goes under the rubric finite patient horizon. So the horizon is just a fancy name to say population size. So, so this is a literature that deals with what do you do when you have a limited population and you want to do some investigation, and if you want to do a clinical trial, how many patients you put into the trial, and then the knowledge gained from the trial patients is then applied to the other patients in the clinic. And so I work with my colleague at Columbia, Ken Chung, recently, and we got a paper published recently in AJPH dealing with this question of how to design implementation studies for quality improvement. So the question we addressed was how do we allocate the sample between the trial and in a sense, the consumption of the knowledge gained. And also, the result was actually really interesting. So when you have a chance, please take a look at that. We took a Bayesian approach because what the local informants know is important. And just very quickly, if the local informants' current knowledge is neutral between the two treatment options, then any local investigation is better than no investigation. And then for the scenarios that we made up with a total population size of 500, the optimal design is to allocate 50 patients to each arm of the trial. So use a total of 100 patients, and then the results of the trial is applied to the other 400 patients. And then the decision rule from this small trial is whichever treatment looks better is applied to the other 400 patients. So that's equivalent using a one-sided p-value of 50% instead of the usual 5%. And then if the local informants have a slight preference for A than B, then the configuration of the local trial changes somewhat. And then when the local opinion, the existing knowledge is strong, then it's better to go with that knowledge instead of doing a new investigation. It doesn't help. So I think it's intuitively very appealing that we can incorporate the local informant's knowledge in an informed way to decide what is useful to do a local investigation or just go with the existing
Now, another
knowledge.
question about that. So if you have a patient population of 500 and you're going to do the study on 100, wouldn't the 100 have to be representative of the 500?
Yeah, well, that's one of the assumptions we're making. Yeah, so
Not everybody may want to participate, so then you're introducing bias.
yeah, so this is a very, I guess, a concept paper. So we just want to see for ourselves, and I think it's a useful demonstration. Under some not totally crazy conditions, is there a way to design the study that's different from the usual way we design research studies? And I do hope that some of our colleagues can take on. I think it's a very important issue to help get a better understanding of the methodology for designing those local investigations. Currently, I think there's very little. And so we usually don't do an empirical investigation. We usually just, I think, go by the informant's current knowledge. But I think I hope that our paper at least demonstrates that there are some situations under which it might be advantageous to do a local investigation, and then a lot more work needs to be done. I think that's about.
This might be useful since we're mostly behaviorally focused folks here.
You want me to say a few words?
Just a couple, yeah.
Okay, so I also collaborated recently with David Moore at Northwestern, and also Ken Chung and several other colleagues at Northwestern on this also conceptual paper called Continuous Evaluation of Evolving Behavioral Intervention Technologies. So that long title refers to the fact that for many of my colleagues who work in technology-based intervention strategies like mHealth, the intervention can evolve fairly rapidly because somebody might come up with a good idea, and then you're moving from 3.1 to 3.2 and then 3.3 maybe in a matter of a couple months. And so it's very different from the traditional research program where we evaluate treatments in a 5-year study, and then we expect that the good treatment will be used for many years to come. But here, the shelf life for those treatment options might be a matter of a few months, and pretty soon something new will come up and will take over. So David's vision is to, in a sense, combine the development and the evaluation at the same time into this CBIT model, which can be interpreted as a horse race. So you have the plausible treatment options and going, and we invite patients to try out those treatments. And then new horses might enter the race if they are considered to be a plausible improvement, but we don't know yet. And then over time, we collect the use data and hopefully also outcome data. And then when we reach the point where some of the horses are falling behind, and we have reasons to believe they are not competitive compared to the other horses, then that horse is withdrawn. And so over time, the herd of horses in the race evolve over time. And there's no final winner. So at any time, we might have several viable options in the race. But I guess the long-term goal is for this paradigm to continue to improve outcomes over time. So the horses get better and better over time, even though we will never have a winner, because anyone that is a winner today will be taken over by some new horses pretty soon down the road. Another possible option is to use adaptive assignment. There's a very nice statistical literature for the static situation. If we have several treatments we're comparing, we can adapt the treatment assignments using interim data. So if A looks better than B, but we haven't reached the final conclusion yet, then we begin to allocate more patients to A and allocate fewer patients to B to reduce the burden on the trial participants. But here, it's like adaptive assignment with a dynamic entry. So the new horses could be coming in to be evaluated along the way. So let me take 1 minute to very quickly. So I would think that the single patient trial has the potential to be a useful tool both for research and also for clinical applications. And whether that expectation is real or not needs to be evaluated in studies like PREEMPT. And the infrastructure issues and other implementation issues. And then what I think is probably is most useful to develop in the future is to think about the local investigations that potentially could inform local decisions. So those are questions that are specific to a clinic, specific to an agency, and in addition to utilizing the local informants' knowledge, there's a potential that maybe we can help the local agencies to conduct a local investigation to try to inform those decisions. And those will fall in the domain of quality improvement instead of research, because we're not producing knowledge for the purpose of applying to other agencies, to other patients, but to address a specific local problem.
Oh, questions? We'll take a few minutes for questions.
Yeah.
To follow up on a question that Afifi started to ask, can we circle back and make some generalizable statements from these individual trials? And what I'm thinking is this really similar to a model with fixed individual effects and truly exogenous change in treatment? And then, and can you think about these improvements or not as a function of the characteristics of the patients, and then apply them to other populations where you would have a better shot at what the first therapy should be?
Definitely, yeah. So this could be a promising way to do research in the sense that we first try to address a local need for the specific patient or for a specific agency, and then in a sense, it's like doing a lot of case studies. And then what we learn from those individual studies definitely could be combined and to estimate how the effect looks like in the aggregate, not only for the overall patient population, but also for subpopulations. So I guess this is what economists call the fixed effect. So each individual patient has a fixed effect, and they might all be different. And so we try to estimate the fixed effect, but at the same time, we can also take a Bayesian approach to combine across patients. And then in the Bayesian framework, we can also use empirical-based methods to try to improve the method for individual patient. So there's a possibility that if for a specific condition, enough single patient trial have been done, maybe we could declare closure, and maybe we know enough to proceed with future patients. Well, I started with, I guess, a criteria for the application of this methodology that there is not such existing knowledge that we could use. But if a single patient trial has been used for this condition for some time, and we have enough knowledge, then that condition will no longer hold. Maybe we can just use that knowledge.
Yeah, it seems like the trick, though, is to make sure that the treatment changes are exogenous. So I think very often we change treatments for a patient because we've moved to treatment B because treatment A failed, and you don't know whether that's also going to influence treatment B.
So this is trying to do it in a systematic way to get good information instead of I guess responding post hoc manner to the treatment response. Yeah, Neil.
Naihua, what do you think about the use of this in sort of in more clinical practice, in the sense that I'm thinking of, I'm taking care of a 13-year-old boy with inflammatory bowel disease, and he's on regular medicine, but he comes to me and says, I really like the tennis player Djokovic, and he went on a gluten-free diet, and I want to go on a gluten-free diet and see if that gluten-free diet's going to make me better. And there's nothing to say one way or another, or I want to try yoga and see if that enhances. Can you use the N-of-1 trial with the individual patient over a period of time and adjust that, and then build that into a learning health system? Because if that individual patient's connected to other patients, and he's connected to 14 other young kids this age, and says, hey, I'm doing yoga now, and I'm not going to the bathroom as often as I was, when I was just taking the medicine on my own. Can be used for exploratory, kind of almost like an artificial intelligence program built into clinical programs.
Yeah, so the key criteria is whether the fact that yoga is pretty spontaneous. So if we do a trial on yoga for this patient, then you would ask him to do yoga for a week, stop it for another week, and then do it again for another week, switch back and forth several times in a systematic way to see whether he's doing better on the yoga weeks than non-yoga weeks. If yoga has a long-lasting effect, if you did yoga for a week, then you're good for 6 weeks, then this might not work. But if the effect of yoga goes away pretty quickly, then this potentially could be a useful way to evaluate that. And I definitely agree. So the clinical aspect of this methodology that I think is potentially very promising is that this can help inform the decision making in clinical care. But then that information could be combined down the road to inform the decision for others. So although the primary objective, I think, is quality improvement and clinical care, the research knowledge potential could be gained as a product in the future. Yeah.
So thinking about behavioral interventions, how do you control for the effect of randomization itself? So let's say like ARB adherence interventions and text messaging. So you try one intervention and then shake it up with another, level frequency or some other modality. But it's maybe even the transference from one to another, or even the washout, that could have an effect on adherence in and of itself.
Yeah. So the key question is whether there is a carryover effect. If there is a carryover effect, like all crossover trials, it might not work very well. But if there is no long-lasting carryover effect, then this might potentially be useful. So you mentioned the frequency. So potentially this could be a way to evaluate what is a good frequency for that specific patient. You can let the patient do it with a high frequency for 1 week, low frequency for another week, switch back and forth. Another thing is I took out the slide for the randomization. So this can be randomized. It does not have to be randomized. As long as the assignment, the switchover, is systematic instead of selective. There might be a lot of advantage to do this in a non-randomized systematic way, like ABBA, that helps control for any possible time trend over time, but does not have to impose a big burden on the end users to understand and accept randomization.
Yeah, you posed this challenge to us, to be investigators, not researchers. What's the story behind
So that's the slides I skipped. So the HHS defines research or human subject research as activities that aim to produce generalizable knowledge. So when we do parallel group trials, usually that's our purpose. We want to learn whether A works better or B, and then apply it to the future patients. But for single patient trial, for clinical applications, not all single patient trials, for clinical applications, the type that Neil was referring to, our primary purpose is the local knowledge. We want to resolve the question for this patient, and any application to future patients is a byproduct. It's not a primary purpose. So in that sense, I think this is more like a quality improvement than the human subject research, the way OHRP defined it. And I think a lot of our work, especially I think the statistical methods, have tended to focus mainly or almost entirely on research. and less so on quality improvement. And sometimes it seems that there is a, what's the word for it? Attitude that quality improvement is not such rigorous. And I think that might be true on average, but maybe not for every quality improvement program. But I think the thing that at least I think biostatisticians have an opportunity to do is to help develop the methodology that's useful for quality improvement to address those local issues. So we don't always rely on the existing local knowledge, but we have a mechanism to produce good quality local knowledge when it's needed. And that, I think, potentially is a very big contribution for statistical methods. And for that purpose, if I want to hold the research strictly to this definition, then we're not doing research. We're doing, I guess, investigation, which is not this kind of research. So in that sense, I hope that we will be enthusiastic to do investigations as investigators and not necessarily always have to consider ourselves to be researchers in this sense. If I can add a word to it, another paper I'm working on with several colleagues is on this notion that implementation, which I think many of us are involved in, should not be just thought of as science, but should also be thought of engineering, because implementation really involves a lot of local adaptation, problem solving, and a lot of those are really engineering questions and not science in the sense of trying to produce some general principle. And an engineering friend of mine from Hopkins told me of a very neat statement among engineers that over the last 50 or 60 years, it's not rocket science, it's rocket engineering. But we talk about rocket science. Yeah, Steve.
So I have several questions. I'm going to limit myself to one, maybe one and a half.
We can talk more later. Yeah.
But one of the questions is, on the flip side of the coin, it may not be research, but is it billable? So if you bring somebody into the clinical situation and you say, we are going to do this sort of an approach, I'm going to move you between this treatment and that treatment. And a chronic disease, most of the treatments are not going to be home runs anyway, so people are going to they're going to come into this with a certain mindset about what's going to happen with them. Is it billable? There is a chapter in that HRQ Users Guide that we discussed financial issues. And the quick answer is potentially yes. There used to be a commercial startup company, I forgot the name, about 10 years ago that tried to do single patient trials on a commercial basis. So what they offered is any clinician who wants to do this trial for her patients can write such a prescription, and they will fill it with the instructions to the patient. And they were able to get some third-party payers to agree. I think there's a code that was created to bill for such services. So it's like writing a prescription, but it's just writing a
code for re-engineering that research.
Yeah. No. But you don't bill for but you don't get to bill for research. So this is a way to bill it. Yeah. But another aspect is I think there might be also a potential to have more allies in the work that we like to do, or whatever we call it, if we, I guess, market ourselves as we're trying to help solve problems instead of we're trying to do research in the ivory tower. And I think there might be a broader constituents who potentially could be engaged in this fashion than to support research.
Okay, so that wraps up Naihua. Thank you very much, Naihua, again. And we'll take a